Okay, I'm gonna start talking now. So wait, is this being recorded right now?
Yes.
Okay, cool. Okay, so today we're going to be talking about Campbell chapters 5 through 7. Campbell chapter 5 is the structure and function of large biological molecules. Macromolecules are polymers, which are long chains synthesized from short monomer units. These monomers are synthesized through dehydration, which is where an H and a hydroxyl or OH group are taken out from the two units. And these units are broken down by hydrolysis, where water is added between the bonds to basically cleave them. So here, there is a picture of this process happening. And then so the first macromolecule we'll be talking about is carbohydrates. So they're commonly known as sugars. Its monomer is the monosaccharide, and the monosaccharide's chemical formula is in some multiple of CH2O. So for example, glucose has the formula of C6H12O6. And so there are two hydrogens for every carbon or every oxygen. So monosaccharides have a characteristic carbonyl group, which is a C double bonded to O, and also hydroxyl groups, which are OH groups. These monosaccharides are joined together by glycosidic linkages into disaccharides, which are two monosaccharide units, or polysaccharides, which are more monosaccharide units together. So there are two main types of monosaccharide rings, which is the alpha ring and the beta ring. The alpha ring has its OH groups on the same side, whereas the beta ring has its hydroxyl groups on opposite ends, as you can see in this little graph here. So these two forms are interconvertible and have approximately the same proportions in nature. So polysaccharides have many different functions, including energy, which is what we think of when we think of sugar. They also can store energy in the form of starch in plants and glycogen in animals. And they can also function in structure. So for example, cellulose makes up a lot of plant cells, cell walls, and chitin makes up fungal cell walls. So carbohydrates are named after its number of carbons. So for example, triose has 3 carbons, since there's pentose has 5 carbons, and hexose has 6 carbons. So in this little picture diagram here, it shows the formation of glycosidic linkages by dehydration. So next, I'm going to talk about lipids, and they are also known as fats. So they are very hydrophobic due to the lack of polar functional groups on its fatty acid chain. So as you can see here in the graph, there are only C and H's on the fatty acid tails, which makes it very nonpolar and thus hydrophobic. So these chains only have these lipids are composed of a glycerol group, which is on the left here, and then different numbers of fatty acids, which are usually 16 to 18 carbons long. So what is pictured here is called a triacylglycerol, which is 3 fatty acids and a glycerol group together. So there are 2 types of fats in general, saturated and unsaturated. So saturated fats have fatty acid CH chains that don't have double bonds, which makes them straight. And these fats are solid at room temperature, since these straight fatty acid tails don't take up enough space to move around enough. And without enough movement, these molecules will remain in place and be solid. So an example of saturated fat is butter or animal fat, which is usually solid at room temperature. And the other type of fat is unsaturated fats, which have double bonds in their fatty acid chain that results in bent fatty acid tails that take up more space. So these are usually more liquid at room temperature, since the molecules take up more space and need to move around a bit more. So an example of an unsaturated fat is vegetable oil. which as you probably know is liquid at room temperature. Lipids can also be in the form of steroids, which contain 4 fused CH rings and are characterized by the chemical groups attached to them. So steroids, in a sense, are not the drugs you hear about, which some athletes might use to grow their muscles, but they are often hormones or signaling molecules. So an example of a steroid is cholesterol, which I will talk a little bit about later. And these are often found in your cell membrane at healthy amounts, but too much can cause fat buildups in your blood vessels, which is not good. So the main function of lipids include energy storage, as you probably know from like animal fat, and also being part of the phospholipid bilayer. Yeah, what's up? You have a question?
Excuse me, so what does the 4 fused CH rings mean?
So steroids have 4 rings of CNH. So it's like the kind of like the fatty acid tail, but there are less carbons and they kind of form rings together.
Okay.
Yeah.
So wait, what are steroids again?
So steroids are signaling molecules like hormones, or they can also be part of your phospholipid membrane. They are also characterized as lipids though. Yeah. Okay. And the other main function of lipids include signaling and being a hormone like steroids. Hold on. Okay. So next, a protein is a complex of many peptide polymers and a specific 3-dimensional structure. So the protein's monomer is an amino acid, which consists of an amino group, which is the NH2 on the left here, a carboxyl group, which is the C double bonded to O and also bonded to an OH, and a central alpha carbon in the middle, and also a side chain that has very specific characteristics depending on what the side chain is. So these side chains can be they can be acidic, basic, nonpolar, or polar, depending on the different atom groups in there. So these amino acids form peptide bonds with each other into a polypeptide, which is the polymer. Proteins have many different functions, which include being an enzyme, as shown here, being a hormone, acting in defenses as antigens or antibodies, structure, motion, and mobility, such as in muscles, being receptor proteins, being transport proteins, and hold on. Okay. And also storage. The protein structure has 4 levels. So the first level is called the primary level, and it consists of just amino acids linked by peptide bonds. So this level structure is very important, as changes in the primary structure can significantly affect protein structures later on, since the amino acid sequence is very specific to each protein and its characteristics. The secondary level is determined by hydrogen bonding interactions between the polypeptide backbones. So like the interactions between the OH group here and the amino group. So they have different polarities, and then that will cause interactions between them. These interactions will fold the protein into alpha helices and beta sheets. So this folding is determined by the amino acid sequence, since there is slightly different structures between amino acids, the alignment of the dipole, which is the polarity, and also specific covalent bond angles that can vary a little bit. So it's important to know that these are that the secondary level folding is not formed because of interactions between the R groups, the side chains here, but it's actually influenced by interactions between interactions in the big polypeptide backbone here. So the tertiary level next is formed from interactions between the R groups, the side chains that I was just talking about. So the amino acids with hydrophilic side chains will tend to stay on the outside of the protein since they like to face the more watery part of the cell or the outside environment. While the amino acids with hydrophobic side chains will tend to stay on the inside of the protein, and on like the interior part, since they don't like to interact with the outside, more watery aqueous environment. There are also disulfide bonds, which are formed between sulfurs, and they form a connection called a disulfide bridge. So in whole, the interactions between positive and negative side chains help to hold the protein together into its tertiary structure. So since tertiary structure proteins have a pretty developed structure, the most basic protein functions emerge at the tertiary level. And lastly, the quaternary structure is formed from multiple tertiary protein subunits. So an example of a protein that is basically a quaternary structure is hemoglobin, which is made of alpha and beta subunits that contain heme, which is a form of iron that binds to oxygen. Any questions so far? No? Okay. I can't see the chat, so yeah. Okay, next. Proteins can be denatured when exposed to extreme pH, temperature, or salinity changes. Remember, proteins beyond the primary structure are not held by covalent bonds, but are instead by hydrogen bonding and other intermolecular forces that are not as strong or permanent. For example, transferring a protein from a polar to a nonpolar environment can drastically alter its structure, since the hydrophobic amino acids would try to flip themselves on the outside, since the outside environment is more nonpolar. And then the hydrophilic, the polar amino acids would try to flip themselves onto the inside, since the outside environment is nonpolar. So to help with protein folding, there is a small protein capsule called a chaperonin, which the protein would enter to have a more ideal environment to help fold properly. Okay, so next is nucleic acids. So the monomers for nucleic acids are nucleotides, which are composed of a 5-carbon sugar, either ribose. Yes, what's your question?
Just a note, for the chaperonin thing, like I think it's this slide because like you just talked about pH and chaperonins and like protein folding, and like it says 5.4 proteins here. And now you're talking about nucleic acid.
Yeah,
So-
because I finished talking about oh, wait. Were my slides not changing? Sorry.
No, when you were doing the protein folding thing, we were
Yeah.
looking at 2 images of proteins.
Yeah. So the chaperonins are on the protein slide though. Just here, right? And then next is nucleic acids.
Yeah, that's
Yeah. Okay. Okay. Hold on. Give me a second. Okay. So the monomer for nucleic acids are nucleotides, which has a 5-carbon sugar, which is ribose or deoxyribose, a nitrogenous base, which is either guanine, adenine, thymine, and cytosine, or replacing the thymine with the uracil, and also a phosphate group. So the pyrimidines, which are one type of nitrogenous base, are cytosine, thymine, and uracil. And on the other hand, purines are adenine and guanine. So pyrimidines have only 1 ring, while the purines have 2 rings. And as you will learn later, a purine always pairs with a pyrimidine to keep the DNA strand at a specific width. So the DNA strand won't be too wide or too narrow. There are also 2 types of 5-carbon sugars, a deoxyribose and a ribonucleic, and ribose. So deoxyribose has 1 less OH group, while ribose has the extra OH group. So, do you have a question or are you just like Marco? Oh, are you okay?
No, I was just doing this.
Okay. I see you on the bottom of my screen. So, and these, the ribose basically differentiates the RNA from the DNA. But also RNA is single-stranded, but DNA is double-stranded. And as I have said, RNA has the ribose sugar, while DNA has the deoxyribose sugar. The DNA's two strands also run opposite orientations from each other, and this is called anti-parallel. And the purpose of nucleic acids are to store and transport genetic information. So DNA basically stores your information, and then RNA can transport your information and allow for translation. Okay, next. Do we have any questions about that chapter? Anthony, what are you doing? Okay. Okay. Next, we'll talk about chapter 6, which is a tour of the cell, where I will discuss the organelles a little bit. So there are two types of cells, prokaryotic and eukaryotic. Prokaryotes include bacterial cells, while most mammal cells are eukaryotic cells. So what are the similarities and differences between these cells? Oh, okay, you want to answer? Yeah, go ahead. I mean, it's already on the slide, but yeah.
Wait,
You
no,
can
we didn't change the slide though.
My bad. Did it change?
Yeah, basically
There's a
they're
lag.
both
I
cells,
don't-
right? And then like both of those cells can make proteins and carry out
Mhm.
basic cell functions. But
Yeah.
then like the main difference is that prokaryotes don't have a nucleus. They have like this nucleoid region that they just scramble out all like they just stuff their DNA in there.
Yeah.
And then also their organelles are less complex.
Yeah, that's true. Well, they're not membrane bound, so they're just like less
They're
of
like,
them.
wait, what?
No, they don't
Oh, wait,
have
the
membrane
nucleus.
bound organelles. So like their organelles aren't enclosed in a membrane. So yeah. Exactly what Marco said. The two types of cells are similar since they have cytosol, which is the cellular fluid. They have ribosomes that can make proteins. They have genetic material, and they also have at least a cell membrane. However, prokaryotes are different since prokaryotes are different from eukaryotes since prokaryotes don't have membrane-bound organelles, and they also have a nucleoid, which is basically a mass of their genetic material just in the cell. And they don't, and it's not separated by a nucleus like eukaryotes have. Okay, also, a side note, there is a reason for why cells are very small. So cells can't really just grow bigger like we do because small cells increases the surface area to volume ratio, like shown in this picture. So this gives cells more, yeah, what's up? You have a question? Aston?
I can't see like the whole slide. Like it only shows me part of
Which part can you not see?
like the like 1/5 of the whole slide. So
Oh, let me try resharing.
Actually, Aston, I had that problem before. You should have just like resized it, and then it would like
Wait, Marco,
automatically.
you could see the slides, right?
Yeah, I could.
Yeah, okay.
I just resized it a little bit, and then it automatically fixed itself.
there we go.
Okay. Okay, anyways, as I was saying, this gives cells more area for material exchange. What is going on? Okay, also small cells take generally less energy to maintain, which is good for the cell. Okay, so here is a list of your eukaryotic cell organelles, which are very important. So your nucleus, it stores the genetic material, your DNA. The nucleolus, which is on the bottom, but it's very related. It produces the ribosomes from the rRNA, from the ribosomal RNA. Moving on, the rough ER has ribosomes attached to it, which are responsible for protein production. The smooth ER, which looks like the rough ER, but it doesn't have the ribosomes. It is responsible for lipid production, and also detoxification. The Golgi apparatus packages materials and vesicles for transport. The lysosome digests particles. The ribosome produces proteins. The mitochondria produces ATP, and then the peroxisome, it basically alters metabolism products and has hydrogen peroxide as a product, which it turns into water. But I'll talk more about that later. Okay, here's some more. The centrosomes are the locations of microtubule initiation. The cytoskeleton includes microfilaments, microtubules, and intermediate filaments, and these reinforce cell structure and also has a role in movement and mobility. The plasma membrane separates contents of the cells from the outside, and it regulates material exchange between the environments. The cell wall, which are not in animal cells, are a rigid outer layer. So actually bacterial cells have a cell wall, so do plant cells and fungal cells. And plasmodesmata in plant cells are tunnels that basically connects two cells and allows for material exchange between the two cells. Okay, I'm gonna go into a little more detail about these. What Ivan, what are you doing, dude? Okay.
Anthony is actively gaslighting middle schoolers, so I'm trying to stop
Anthony,
that.
stop. You know, I got yelled at because you were being disturbing.
I'm just saying, I posted the link.
Okay.
Okay,
I'm
so
gonna continue. Okay. Anyways, the nucleus is where the DNA is stored. It is surrounded by a nuclear envelope, which has a pore complex of small pores that allows entry or exit of materials such as mRNA. This DNA in the nucleus is organized into chromosomes, which is DNA wrapped around many histone proteins. So this DNA and histone complex is called chromatin, which condenses before cell division. Ribosomes are synthesized in the nucleolus from ribosomal RNA, and they exit the nucleus to carry out protein synthesis. So ribosomes can be free or bound to rough ER, but their products have slightly different endpoints. So free ribosomes produce materials that are usually for use within the cell. On the other hand, bound ribosomes on the ER synthesize materials that are usually transported into another membrane-bound organelle or out of the cell because it is very nearby to the Golgi apparatus, which is responsible for transport. The endomembrane system consists of organelles, which are largely made of membranes. So the smooth endoplasmic reticulum, also known as smooth ER, in the system detoxifies drugs and synthesizes lipids like oils, phospholipids, and hormones. Basically, you can think that a cell that has to process a lot of toxins, such as liver cells, would have a larger number of smooth ER because there's more toxins to process. The rough ER has ribosomes that produces proteins for extracellular use, but also can produce more membrane for the endomembrane system to grow. The Golgi apparatus receives and transports substances out of the cell through vesicles that it pinches off itself. Lysosomes digest macromolecules such as food or other wastes or damaged organelles, and it has enzymes inside of it that can do that. The lysosome is pretty useful, but it can also cause cell death when lots of them break at the same time because of the digestive enzymes inside of it. Finally, vacuoles can transport solutes or store substances. Examples of these are food or contractile vacuoles, which move excess water out of the cell. Plants also have a very huge central vacuole for storing water, which is good for the cell. Okay. Next, I'm going to talk about mitochondria and chloroplasts. So the main function of mitochondria and chloroplasts is to change energy from one form to another. What does that reaction do? Okay. While mitochondria change sugars into usable energy, chloroplasts use light energy to make sugars. So the mitochondria and chloroplasts could have come into being through the endosymbiont theory, which states that these organelles could have originated as small prokaryotes, which were then engulfed by bigger cells. The independent genetic material, the double membranes, and self-replication all indicate that this theory is somewhat provable, or is a good theory to believe, I guess. The mitochondria is in both plant and animal cells and kind of looks like a hot dog. It has a smooth outer membrane and folded inner membranes called cristae, which give more surface area for cellular respiration processes. The mitochondrial matrix on the very inside is enclosed by the inner membrane and consists of a lot of enzymes relevant for cellular respiration, as we will talk about later. Chloroplasts, on the other hand, are found only in photosynthetic cells like plant cells. So chloroplasts use light energy to to basically make sugars. So there are stacks of thylakoids called granum inside the chloroplast membrane, and the fluid outside of the granum is called the stroma. And these regions are very important during photosynthesis. Next is peroxisomes. They remove hydrogens from metabolic products and then transfer them into oxygens, which generates hydrogen peroxide, or H2O2. So hydrogen peroxide is usually toxic for the cell, but peroxisomes are compartmentalized, which means they are separated from the rest of the cell environment by a membrane, so that prevents the peroxide from getting out. And it also contains enzymes that can turn the peroxide into harmless water, which is not of harm to the cell. Next is the cytoskeleton. So the cytoskeleton provides mechanical support for the cell mostly, but it could also function in cell motility through interactions with motor proteins. So here on the upper right, there is a red little motor protein that can bring a vesicle and basically walk itself along a microtubule to bring materials to where it needs to go, which is pretty interesting. The cytoskeleton is composed of microfilaments, which are the smallest, intermediate filaments, which is in the middle, and microtubules, which are the biggest. So microtubules grow out of centrosomes and mostly help with motility in cilia or flagella, which are little, I guess, like, so the flagella is like a whip that basically moves like this and moves the cell along, and the cilia are like shorter and more numerous versions of that, I could say. Anyways, intermediate filaments, which are smaller, make up the permanent framework of the cell and also help reinforce organelle positioning. And then microfilaments, which are the smallest, are also known as actin filaments and have functions in bearing weight and pulling forces. So just something interesting, actin and myosin filaments together lead to contractions, which is how muscles contract as well. Also, I don't know if you guys need to know this, but those two together, microfilaments can form the semi-solid cortex on like the outer part of the cytoplasm. So there are also extracellular components outside of the phospholipid bilayer. Yeah.
Wait, no.
Okay. The cell wall exists in bacteria, plant cells, and fungal cells. to protect the cell, maintain its shape, and also to prevent excessive uptake of water in plants to make sure that they don't explode. These cell walls are mostly made of cellulose in plants or chitin in fungi. Animal cells also have an extracellular matrix which consists of glycoproteins, which are carbohydrates attached to proteins. These bind to integrin proteins, which then help to relay information from the extracellular matrix into the cell. So finally, there are a variety of cell junctions between cells. Plasmodesmata in plant cells are like tunnels between the cells that allow cytosol and other material to pass through, like I have talked about previously. Tight junctions in animals, which are shown here, prevent leakage of materials, and desmosomes in animal cells, which are here, serve for anchoring and cell-to-cell attachment. And lastly, animal gap junctions are kind of similar to plasmodesmata, as you can see here, and they are also like little tunnels between animal cells that allows for content exchange. Okay, we finished chapter 6. Do we have any other questions or like thoughts on the chapter? If not, I'm gonna stop sharing for like about 5 minutes so we can all take a little break and like process the information. Okay. Damn, okay. Well, guys, stop. Oh yeah, we're forming teams, so you might wanna know some stuff about this. Anthony, no, you're literally. If you guys get Derek on his team, you're gonna like do so good. Yeah. Okay, guys, stop talking. Let's like. Okay, I wanna reshare. Oh my gosh, hold on. Thank you, Marco. Okay. Okay, moving on. Talking about membrane structure and function in cells and in general. Okay, so the lipid membrane is made of phospholipids, which contain one phosphate group attached to two fatty acid tails. So these phospholipids are amphipathic, meaning that they have both hydrophobic and hydrophilic ends. So the phosphate group is polar, while the fatty acid tail is nonpolar, and therefore phospholipids will orient themselves in a bilayer with the phosphate heads on the outside where the water is, and then the tails on the inside where the water is not. These phospholipids are quite fluid, meaning that they can shift easily within the membrane. And in fact, the membrane is supposed to be quite fluid. So cholesterol, which we discussed before, and as you can see here, is a 4-carbon chain with like little thing on the top. Those cholesterol often exist embedded in the membrane to maintain the fluidity of the membrane in cold temperatures. Okay. The phospholipid membrane is a fluid mosaic model because of the many proteins dispersed in the membrane like a mosaic. There are two main types of membrane proteins, which are integral proteins and peripheral proteins. Integral proteins penetrate through the lipid bilayer and has both nonpolar and polar sections. So the nonpolar sections are going to be in like the middle part where it touches the inside nonpolar part of the membrane, while the polar parts of the protein are going to be on the outside. So the other type of protein is peripheral proteins, which are loosely bound to the bilayer and are generally polar since they can be on either side of the membrane. And in addition to these proteins, there are often carbohydrates bound to lipids, which are called glycolipids, and also glycoproteins, which we just talked about, which are carbohydrates bound to proteins, and these help with cell identification. Okay, the phospholipid membrane, because of its nonpolar and polar properties, is selectively permeable. So this means that the cell membrane, which is hydrophobic on the insides, only lets small uncharged or nonpolar molecules through, like oxygen gas or carbon dioxide gas. Water can also diffuse into cells because they're very small, but very, very slowly because they are polar. So the cell solution to this is adding transport proteins to the cell membrane. These transport proteins are needed for large or charged molecules, and they are able to bring these molecules through. So some of these proteins can be channel proteins that act like a tunnel for small charged molecules, such as aquaporins for water, which allows for a faster diffusion of water. So in summary, the selective permeability of a membrane depends on membrane structure and transport protein presence. Okay, so there are two types of transport of materials, which are passive and active. So during passive transport, the proteins diffuse from areas of high concentration to areas of low concentrations, since these molecules want to have more space. So as you can see here, the dye is very concentrated on one end, and then it moves through the membrane and reaches an equilibrium where the concentrations are the same as it moves from a higher concentration into a lower concentration here. And this transport is passive because it does not require extra energy input, since these molecules just kind of move themselves naturally and don't need to be pushed. So specifically, the passive transport of water is called osmosis. This diffusion of water from a higher goes from a higher solute concentration, which is the substance dissolved in the water, to a lower solute concentration. So essentially, you can think of it as water traveling from areas of a higher water concentration to a lower area of lower water concentration. So as you can see here, there are more solutes on this side and less solutes on the left side, and then the water will try to move onto the right side to make the water concentration more equal. So in osmosis, the tonicity of the environment determines the direction of water flow. So when the environment of a cell is isotonic to the outside, that basically means the extra and intracellular concentrations, solute concentrations are equal, and there is no net water flow because there is really no reason for the water to keep flowing in and out. There will be water flow, but they will be approximately in equal amounts. So this is normal in animal cells, but in plant cells, the cell is slightly shrunken, and this is called flaccid. When the environment of a cell is hypertonic to the outside, that means the extracellular solute concentration is higher, and then there is a net flow of water outside. Yeah, Aston, what's your question?
What would happen? What would happen if wait, no. Wait, I need to think about a question first.
Okay. Should I come back to you? Or
Yeah, you can come back to me later.
okay, just tell me when you're ready, okay? Okay, anyways, so in a hypertonic environment, there is a net flow of water outside of the cell, which is seen on the right side here. If you can, yeah, okay, Aston, are you good now? Or okay, Isaac, I see your hand up though.
Yeah, what's really the difference between like the like what's the cell below the red blood cell? Like I don't know which one that is.
Oh, those are plant cells. So we can see they have an outer cell wall. And there is a huge central vacuole here, which is mainly for storing water. So on the top is an animal cell, which is a red blood cell here, and then the bottom are just plant cells. Mhm. Okay, Austin, are you ready or?
Yeah, I'm ready. So my question is that what about humans? Like if this happened to a human body, like would the body explode, or would it just have like heart disease?
Cells do start bursting, yeah, that does happen. So I guess if you just dunked a person in water for way too pure water without any solutes, basically deionized water, then your cells will start popping. Yeah. Okay, Marco.
So first of all, a note to Isaac. I think you should distinguish the plant cells by looking at the chloroplasts because that's one of the main distinguishing features.
Yeah.
But actually, my main question was, what makes it so that when a human blood cell, when a human cell or an animal cell will get lysed, but plant cell, it's actually the ideal conditions. Why?
So the plant cells have a very hard outer cell wall. So that basically prevents the cell from bursting or lysing. So also the plant cells have a really big central vacuole which can store a lot of water. So it's I guess it's healthier because the cell can take up that much water, but the animal cells cannot because they don't have that cell wall to protect them.
So then, hold on. But then why would it be flaccid when a human cell will be normal?
I don't know the exact reason of why this is considered normal for plants per se. I can check that out and I'll answer it next meeting.
All right.
Okay. Or do you still have Aston?
Somebody tell me what's going on in chat.
It's not your business, simply put.
Okay. Let me just finish.
It's not any of our business. I'm sorry for being rude, but like,
What?
yeah,
Okay. Thank you.
that
Mark.
is quite literally, yeah.
Okay, so and then when the environment of a cell is hypotonic to the outside, that means the intracellular cellular concentration is higher, and then there will be a net flow of water into the cell. And then this causes animal cells which don't have a protective cell wall to burst. But this is what healthy plant cells should be like, since they have a very hard, rigid cell wall that which is basically what I just said, to prevent the cell bursting. And then we call this as a turgid plant cell. Yeah. Okay. Charged or polar molecules, as we have talked about before, are unable to directly diffuse through the cell membrane, since the membrane is very non-polar on the inside. So then these charged or polar molecules have to be helped through the cell membrane through channels. This is called facilitated diffusion, since the molecules are still moving from a higher to lower concentration, but they are being facilitated across the membrane. So these channels act as like a little tunnel for these ions or charged molecules to flow through, and many of these are actually gated, which means they open or close in response to a stimulus. So the other type of transport is active transport, where the substances are moved against a concentration gradient. This requires energy to be provided as ATP, because the substances don't want to be moving the other way around. An example of active transport is the proton pump across the mitochondrial membrane during cellular respiration, which we will talk about more next week. Also, active transport can be coupled with passive transport, since the diffusion energy provided by the passive transport can be used to power the active transport of another substance, and this is called co-transport. Yes, sir.
So basically those proton pumps just basically kind of like pump things out.
They move the protons from a lower gradient to a higher gradient. And then we'll talk a little bit more about this later, but then the higher gradient protons will then diffuse back through the ATP synthase, and then that creates more ATP. Yeah. Okay. So materials can also be transported into or out of the cell in bulk. So exocytosis is the bulk expulsion of materials, in which a vesicle fuses with the cell membrane with the material it needs to like remove, and then it releases its materials once it fuses with the cell membrane. So endocytosis is the bulk intake of materials, and there are 3 types. So so this is generally done by the cell membrane pinching in and forming a vesicle, and then taking that back in. So phagocytosis, which is also known as cell eating, is the intake of food particles or pathogens. Penocytosis, which is basically cell drinking, is the intake of small molecules or minerals. So finally, receptor-mediated endocytosis involves the binding of surface proteins to specific ligands, which are then taken in by the cells. Okay. That is all I have for you today. Do we have any questions or anything you want me to go over again? Okay, if not, I will stop sharing, but I have some questions that are good for review. And we have 4 minutes left, so I'll just ask you guys some of those questions. So, and these questions will not be displayed on text because Bible is like read out loud, so yeah. So what kind of linkages connect glucose monomers in starch? You can put this in chat to me, direct message me this, or like raise your hand and I'll tell you later. Okay. I'll repeat this once. Hold on. What kind of linkages connect glucose monomers in starch? Oh, also, Marco, to answer your question, so having turgid or like really big plant cells is normal because the plant cell being bigger allows the plant to basically grow bigger and like expand.
Okay.
Yeah.
Wait, am I supposed to raise my hand in this question?
Or actually just message me, like direct message me the answers. Guys, I need more answers. Come on. We'll be picking teams soon, so I do expect you guys to know stuff like this. Because
Would-
I also just said it. I know, Marco, you answered. Very good. Okay. You don't have the questions? Yes, because I'm not going to be displaying the questions. Yeah. Because during Bible they don't display questions either. They just like okay. Anyways, the answer to that is a glycosidic linkage, which how do you access them? I'm sorry. Anthony made these questions, so there's no like access place, I guess. Yeah. Seems like people have it. They don't. Trust me, they don't. Okay. So these linkages are glycosidic linkages, which I have talked about in the slides. So cool. Okay, next, I'll go like a minute or two over time, but this is in like Bible format. Okay, so here's the question. Is ribose an example of A, a hexose, B, a triose, C, a pentose, or E, an octose? E? D, no, D, an octose. Okay, please message me the answers.
Yeah, why don't you do like XY's XYW?
It's okay, it's okay, it's okay. Okay, I'll repeat one more time, but it's better for you not to have to repeat. Okay, so A, hexose, B, triose, 3, C, pentose, and D, octose. Okay. Yeah. I'm not gonna repeat the question again because they don't like the actual thing doesn't repeat a lot. But okay, I'm gonna explain the answers. For those of you who put C, you are correct because it is a pentose. Okay. Anyways, the question was is ribose an example of a hexose, triose, pentose, or an octose? So a ribose has 5 sugars, which is what I was talking about Ivan, what's your language, dude? We have little kids here.
Don't worry, I yelled at him in the chat.
Good. Um, okay. So as I have said, ribose is a 5-carbon sugar, which is called pentose because pent, like PNT, stands for 5, and it's good to know. Okay. Okay. Anyways, we are out of time for this meeting. It was really great seeing you guys all here. Thank you for your participation. I'm proud of you. Okay,
Thank
next week,
you.
we'll be talking about
Bye, everybody.
cellular respiration and energy. and everything.
Bye.
Bye.
Not a swear next time. Thank you.
Yeah. Yeah.
Bye.
Bye. Have a great time.
Bye.
Goes